everyone. I'm Dr. Gurjeet Kaur, and in this video we would be discussing about starch hydrolysis by microorganisms, mainly bacteria. I've tried to make this video as comprehensive as possible. So before we start this hydrolysis experiment, quickly let's see what are exoenzymes. These are the enzymes which are secreted by living organisms, not inside the body but in their external media to work on the substrate, which are very big and which cannot enter the cell. So these exoenzymes, they are usually hydrolytic and they break down these large biomolecules found in the surroundings to break them into smaller building blocks which can be easily used as a nutritive source for the cell. And one such example of big biomolecule usually found in the surrounding is starch. And to break down the starch, a very important enzyme is known as alpha amylase. So this enzyme, it works on starch, and with the help of water it does the lysis of starch and it breaks down the starch into smaller molecules which are mainly a disaccharide which is maltose, a monosaccharide which is glucose, and few dextrins. So this starch will be broken down into maltose, glucose. And as we know, a very important experiment for the presence of starch is iodine. Iodine very quickly reacts with the starch and it gives a blue-black complex. And if there is no starch, no blue-black complex would be formed. So let's start this experiment of starch hydrolysis by bacteria. So for this, a very important requirement is a starch agar plate. Now what is this plate? It's a normal agar plate, but along with that 1% starch is added, which is called as a starch agar plate. And in this plate, roughly we will divide this plate into two halves. In one half, we would be streaking that bacteria, which we know is secreting alpha amylase, which is amylase positive bacteria, and on the other half we would be streaking with the amylase negative bacteria to see how this reaction goes on. Now after the streaking, we have to keep this plate in the inverted position, incubated for 48 hours. After 48 hours, we will observe bacterial growth on both sides of the plate. Now we would be adding iodine solution, grams iodine, normal grams iodine solution, generously on this agar plate, and will wait for 10 to 15 minutes for this reaction to happen. Now those bacteria which are alpha amylase positive, very quickly during these 48 hours, a lot of alpha amylase as an exoenzyme will be released from their body, and this alpha amylase will work on all the starch found in the external agar plate, and it will hydrolyze that starch and it will convert it into glucose and maltose. So in the surrounding area of this bacteria, all the starch would be hydrolyzed into glucose and maltose. But on the other half, that bacteria which is amylase negative, no amylase as an exoenzyme will be released by that bacteria, and all the surrounding area of that bacterial colony, all starch would be as it is available. Now what this iodine will be do? It will react with all the starch available on the plate. So only that area where there is no starch, no blue-black complex should be ideally found, and on the rest of the plate it should turn all blue-black. So this is what we can see in the result. The whole plate will turn blue-black except for that streaking area where the amylase positive bacteria is usually there, and around that uh, amylase positive bacteria, the whole area surrounding that bacterial colony, it will be not blue-black in color. Rest whole plate will be blue-black in color. So very good example of one such amylase positive bacteria are bacillus bacteria. These are amylase positive and they give a very good positive result for this experiment. And negative reaction or the negative result is usually given by many bacteria, and one such good example is E. coli. It is amylase negative bacteria giving a negative result for this starch hydrolysis experiment. So I'll quickly summarize what we are doing here. We are looking at those bacteria which are capable of releasing alpha amylase as an exoenzyme in the surrounding area, digesting and hydrolyzing all the starch available in the surrounding. And after adding iodine, we can clearly see if starch has been hydrolyzed. If yes, it means no blue-black color around that bacterial colony, and which is a proof that yes, these bacteria are releasing alpha amylase as an exoenzyme in the surrounding. So this was all about starch hydrolysis by microorganisms, mainly bacteria. Thank you.